Okay, class, let's recap what we have done for the steps in artificial selection, and then we will see some examples, right? So, human act as a selective agent or selection pressure. Shh. Who's still talking? Human choose both parents with the desirable characteristics, and we cross the parents to produce the offsprings, and then we select offsprings with the desirable characteristics of both the parents, and then we repeat the selfing and selection for 6 to 7 generations in order to increase the allele frequency, the desired allele frequency, and also to increase homozygosity in the population. So we'll look up to here already. And we have also talked about outbreeding enhancement or hybrid vigor or heterosis. This is to overcome the inbreeding depression. So after we have done all the 6 to 7 generations of inbreeding, then we do one outbreeding enhancement to resolve this inbreeding depression problem. Now you see that number 7 we have not covered, right? So we're going to cover number 7 today. Okay. So now we are ready to look at it. 3 examples of selective breeding. So the first one is introduction of disease resistance. That means resistant to fungus or resistant to bacterial infection for wheat and rice. Because these are the staple food for us, right? Rice is staple food for us. Staple food means the food where it provides you with the most energy every day. We eat most of it is from rice, okay? Wheat is for Western country lah. So number 2, inbreeding means you do selfing, right, for 6 to 7 generations, and then you do hybridisation. That means outbreeding to produce vigorous uniform varieties of maize. Then number 3 is improving milk yield of dairy cattle. So this is very interesting one. So first one is selective breeding to produce disease resistant rice, Oryza sativa. So we know that when we do selective breeding, there are a lot of alleles that we will not select, right? Because we only select the alleles that we want. So those alleles that are not selected will actually disappear, isn't it? It will disappear from the whole population. But we know that those alleles are very important for the survival of the species in a changing environment because it may be useful later. It may not be useful for us, but it may be useful later for the plant to survive. So we want to keep all the alleles that are already there in the rice plant. So the International Rice Research Institution in Philippines, very famous, this one. It holds the rice gene bank. Gene bank means they keep all the different variety, different type of rice from all over the world to ensure that all the different alleles are kept here. Like all your money are kept in the bank. These all the alleles are kept here. All the genes are kept here. Okay? So it's kept here. And then not only that, they also do research to improve the quality and the yield of those plants. Why? Because human population size increasing exponentially, right? So we need to be able to plant those crops that can grow very fast, that are resistant, and that can have high yield. So we have to keep on doing research to improve on that so we can feed the ever-increasing population size. Okay, now they're also producing this what do you call it? Fake meat, right? In US, they are producing fake meat, right? Okay. And we should actually taste that meat also. Okay. So there's a research being done on bacterial and fungal resistant rice. So they do a lot of research to produce those disease-resistant rice to improve its quality and yield. Okay. For example, this one here is showing you the leaf. The leaf is being infected by bacteria, showing you some dried off lines, isn't it? Okay, so this is called the various spots and smuts on the leaf because of bacterial infection. Then the next picture shows you the infection by fungus. Okay, this is infection by fungus. Also looks like rusty spot on the leaf also. Can you see that? So now you know that whenever you see the leaf in this kind of thing, you know it's infected already. Or else it'll be very nice green color, isn't it? Okay. Okay, so that is only the example that you learn for this disease-resistant rice is produced by selective breeding. So this is only example, okay? Now the next example is we want to produce maize, vigorous uniform maize variety through selection, inbreeding, and hybridization. So we know that first we have to select the parents, right? Having the characteristic that we want. And then we cross the parents and get the offspring. And for offspring, you will select offspring and do inbreeding. Inbreeding means selfing, right? Okay. So we cross the offspring with itself or with closely related individuals. And then we choose offspring with desired characteristic and continue to do the selfing selection for 5 to 6, 6 to 7 generations. My generation spelling wrong, huh? Okay, generations. Okay, change now. Generation. Your generation, that correct or not? Huh? I see my slide. Some of them say 5 to 6 generations. Some of them say 6 to 7 generation, right? I will check again and inform you which one is the more accurate one, okay? So after that, we will produce homozygous, genetically uniform offspring. All of them are homozygous, okay? And what is the advantage of them being genetically identical? I mean, homozygous is they need similar environmental condition to survive. And when the environmental condition remain the same all the time, they will survive very well. Okay. You have chosen them to be able to survive very well, right? So if the environmental condition remain the same, they'll survive very well. Okay. And then they'll produce similar quality of crop. But the problem is inbreeding depression. If you keep on doing selfing, then it will allow the deleterious recessive allele to become homozygous. And then the plant becomes less vigorous and less fertile with generations. So that's the problem. So because of the problem, you have to do a hybridization or outbreeding enhancement or heterosis or hybrid vigor, which is a solution to inbreeding depression. So we cross the parents chosen from 2 favorable inbred lines. That means these 2 parents, right, already being produced after 6 to 7 generations of selfing and selection. Okay, they're all pure breeding, but from different farm. Okay, so you bring them, they're all pure breeding with contrasting characteristics. You cross them to produce heterozygotes. So these heterozygotes have got dominant allele to mask the effect of the recessive deleterious allele. So this is the solve problem of inbreeding depression. So now offsprings are now vigorous again, and it show more desirable traits. It means the size is bigger, the yield is better than both the parental line, which could be experiencing inbreeding depression. So this picture shows example. The maize plant on the left and right are purebred. Okay, they are purebred. Purebred means that they are pure breeding. Okay, homozygous from different areas. So when we do a cross between these 2 plants, the offspring plant seems to be taller, right, and stronger than the original parental plant. So this process is called heterosis or outbreeding enhancement. We improve the quality of the plant by masking the effect of deleterious recessive allele. So this is called outbreeding enhancement. Okay. Okay, now we're going to look at selective breeding to produce high milk yield in the cattle. This is very interesting one, huh? So the organism involved is Jersey cow, and the desired characteristic is high milk yield. Now, do you understand what the diagram is trying to show us? When we selectively breed cows that can produce more milk, that means to say that we can raise less cow, but we can get more milk, right? So this one shows us that in 1944, you need 5 cows to get this volume of milk, but now you only need to raise 2 cows and you can get even more milk than 1944. So this is the advantage of selective breeding of cows that can produce more milk. So what is so good about this is not only you raise less cow, but you get more milk, but less cows means what? Less cows means the CO2 released by them to the air become less. Because now every government are actually counting the carbon released from the country, right? From all the activities in the country. So the more cow, the more CO2. You know what the CO2 air do to the air, not CO2 air. What does the CO2 do to the air? CO2 is what? Huh? It's a gas. What does it do to the air? Why is it the government keep on saying we want to reduce CO2, so we want to use an EV vehicle, electric vehicle. We don't want to use the fossil combustion vehicle. Why? Why is it CO2 not good? Huh? Yes, increased temperature. CO2 gas surrounding the earth is like a heat blanket. It's a blanket. Whenever the sun heat, when the heat from the sun shines onto the earth, the light will be reflected back to the air. So when it's reflected back to the air, because of this CO2 will absorb the heat, and then it will heat up the whole earth because it's like a heat blanket. Because this gas is surrounding the earth and this gas is heated up. So the whole earth becomes very hot. So it causes greenhouse effect or global warming. It's because of this. So they want to reduce the CO2 because global warming is very serious. It's causing a lot of iceberg to actually melt, and then the sea level will rise already. So a lot of all those cities or town near the seaside will be flooded. Okay, because of that. So we have to try to reduce that because it's affecting a lot of organisms. So if we can raise cows that can produce more milk, we raise less cow, we have less CO2, but we still get enough milk. So that's the advantage. Okay. So how do you do selective breeding? Female parent, very easy to choose as long as they produce a lot of milk, right? Then we know that they're superior female, correct? So very easy to choose. But father, how to choose? Father don't produce milk or not. How to choose a father that can carry gene that code for high milk yield? Cannot choose father, right? So you cannot choose father, you choose what? You choose a daughter. That means the father's daughter among all the daughters means the cow, not the daughters. Which cow produce more milk? If all of them produce a lot of milk, that means this father also carry high milk yield gene, right? The superior gene. So for father, we have to select father by progeny testing. Progeny means offspring. We choose the offspring for high milk yield. So we look at the performance of the female offspring. Then we cross both superior cow and the bull and to produce the offsprings. Then after we produce the offsprings, we have to select them. So how do we cross them? Of course, to cross animal, we need the sperm and we need the egg, right? So for the bull, we actually collect the sperm from the bull, and then we artificially introduce the sperm into the uterus of the female. So this process is called artificial insemination. That means we inject the semen that collected from the bull, the superior bull, to the superior cow. So after that, fertilization will happen inside the uterus, inside oviduct, whatever. Then after that, embryo will grow. So in this morula stage, morula means a ball of cells, which is embryo in a ball cell stage. In this morula stage, those embryo will be flushed out, like you flush your toilet. Okay, if you flush out from the uterus of the superior female. How did they flush? They use water to flush. They put a tube and then connected to water source, and then it continually be transferred into the uterus, and then another tube for it to come out. Then you collect whatever that comes out. Okay. So after that, they will split every embryo into 2 or more so that they can produce more embryos. So at this stage, I told you before that this stage, the cells are not differentiated, right? So when the cells are not differentiated, no matter how many times you split them, each of the new embryo can grow fully into a new organism. So this is how they multiply the number very fast, isn't it? So after that, now because when the cow is pregnant, I think can only carry one, right? Can only carry one fetus, just like human normally. So, but they want to produce many offspring at the same time. So they have to take all this embryo and insert it into different foster mother so that you can produce more cows, right, at one time. So therefore, they introduce those embryo into foster mother. Of course, not one, many okay, many foster mothers. Okay, so this is the whole process. So let's look at the diagram. Step number 1, sperm is collected from superior male. So how do they collect? You see, this one is not very clear, but actually you can see this is called the dummy cow. So they take the bull and put it on top of dummy cow, and then the bull will start to ejaculate. So they have put a tube there and then collect all the semen during ejaculation. So this is how sperms are being collected. Okay. Then after that, they will do artificial insemination. So these two pictures shows artificial insemination. So what happened is that you can see that this man's hand, right, is totally inside already. Inside where? This one is actually, he puts his hand into the anus and to the rectum. So this inside the rectum, okay? And then this one is the vagina. And then this is the cervix. And this is the uterus. So a tube, which we call catheter, a tube is inserted through the vagina all the way into the uterus. And why this hand has to be here? Because it has to push the cervix to open it to allow this catheter to go in, or else you cannot push the catheter in, because the nature of how the tissues are being arranged, right, is like a valve. A valve will open one way and not the other way, isn't it? So you want to insert the tube in, actually cannot. So you have to push this cervix to allow this to go in. So that's why you do it this way. So this is artificial insemination. Okay. So after that, hopefully fertilization will happen. And after that, they flush out the embryo. Okay. So embryos are flushed out already, and they will be examined for defects like genetic screening, for example. All right. And it can be sex as well, to obtain female offspring. So they can choose only embryo that will grow into a cow, the female. Okay. So embryo can also be split to induce twinning. Okay, so you can get twins. From one embryo can get 2 cows. So after that, embryos are transplanted into surrogate mother. Surrogate mother or surrogate female means foster mother, not the original mother of the embryo. Original mother of the egg, no? So if you want to do that, you must make sure that you have to prepare this female to receive the embryo. Because for all the female, for mammals, they have got this called the menstrual cycle, right? Menstrual cycle. That means their uterus will change in cyclic manner. So certain time of the cycle, it is receptive and it can receive the embryo. Certain time of the cycle, it cannot receive the embryo one, right? Because of menstruation. So we have to prepare the uterus of this surrogate mother so that its uterus cycle will be similar to the uterus cycle of the superior cow. So that when the embryo is ready, the uterus is also ready to receive it, so it can be implanted into the wall of the uterus for it to grow further. So how do they do that? They have to inject hormone. So to inject hormone to the female cow to control their menstrual cycle, to be similar to the original superior female. Okay. So after that, when the offspring grow already, then they have to actually select, right, the offspring with high milk yield. So that become our superior offspring. Then after that, you have to do a selection, inbreeding, selection. Can we do selfing or not? Can we take the cow and do selfing of cow to produce more cow? Can you cross with yourself? Cannot, right? So for cow, we cannot do selfing. So we do inbreeding. Cross it with a closely related individual, right? So we continue the selection, crossing, selection, inbreeding until most of them are homozygous. So again, same thing, 6 to 7 generations. Okay. So over 6 to 7 generations, the allele frequency of the allele conferring desired characteristic will increase, right? After we do selection, inbreeding, selection, inbreeding for 6, 7 generations, the desired characteristic will increase in the population. The allele frequency of the alleles conferring characteristics not desired by the breeder, in many cases, will be lost entirely. So whatever alleles we don't choose might be gone forever because we didn't choose them. So this is a problem here. When it's lost, the variation will decrease, right? Hello? VP is equal to VG plus VE, right? When VP decrease, is because VG decreased, right? We have lost some of the alleles, so the VG has dropped, so VP will drop. So you already learned, right? Variation means VP equals to VG plus VE, remember? Whenever we talk about variation, it can be variation in phenotype, variation in genotype, variation in environmental factor. This one is variation in phenotype, decreases because the raw material means the VG. You can write that in your notes there so you understand. The raw material in this case means all the different type of alleles. All the different alleles, we have lost some of them because we didn't choose them. Okay, so decrease in the allele for natural selection. Just like, for example, I told you the example before. If all the snails have been chosen are all gray shell, one day suddenly flooding occurs, right? And then after that, the cement is gone, it's covered by mud, okay, and the color is different. Then if all the shells are gray color, that means all of them cannot survive, isn't it? Because they're easily spotted by the bird. So this is what I mean by when variation decreases, the raw material that's there for natural selection will actually decrease, so that whenever there's a change of environment, the original favorable allele is no longer favorable. Whatever the rest are favorable, they're already lost. Already lost, right? So therefore, they cannot survive so well and they cannot produce so well in the changing environment. So there's a decrease in the chance of having a favorable allele coding for favorable phenotype to survive and reproduce in the new environment that's different. Okay, so that's why it's so important for us to make sure that even though we do artificial selection, we must keep all the genetic variations that are already there in a gene bank. That's why gene bank is very important. So that in future, we can always go back there and look for allele that confer survival in the new environment, okay? Especially when there's disease, I would say. So let's look at problem. Problem in selective breeding of dairy cattle. What's the problem? Animal takes a long time to reach maturity. So you got the offspring already, okay? So after the embryo grows into fetus, and then after that it's being born, so become the baby cow. Takes a long time for them to grow to maturity and then to be able to produce milk. So you have to wait very long, right, before you can choose the offspring to see that or whether they are high milk yield or not. And number two, long gestation period. Human 9 months, cow longer than 9 months. Hey, you have to wait for 9 months plus for the offspring to be produced or not. And then someone you don't know what offspring is high milk yield or low milk yield. So it's a long time, takes a long time. Then each cow produce one offspring. So the number of offspring produced is very few. So again, it looks like it's very time consuming, it's very expensive, right? This process. You can see not? Time consuming, expensive, because you have to feed them to grow, no? When you feed them, these are all money, right? Okay. Number four, bulls breeding value in carrying high milk production allele can only be accessed indirectly by progeny testing. Means for father, we cannot test them directly. We have to wait until the daughter grow up already and produce milk, then only we can know. So again, it takes a long time. Number five, inheritance of background genes from both parents and suitability for the adaptation of organism in the immediate environment must be considered. What does it mean? For example, we said, oh, New Zealand cow produce very high quality milk and then like a lot of milk. Let's cross the New Zealand cow and Malaysia cow. Can or not? After you cross already, you produce offspring. This offspring, yes, got high milk yield gene, the superior gene, but too bad. This offspring, right, got very long fur. No? You know long fur? Longer, because why? Inherit from the New Zealand cow, right? So can survive in Malaysia or not? Cannot. To make them survive, what must you do? Supply aircon. Aircon cheap or not? 24 hours aircon, no. Freezing cold enough? Cannot. Okay? So very expensive. So we said that we need to consider what? Inheritance of background genes. That means the genes that allow them to survive in local environment. Okay? You must make sure that they can inherit the background genes from the local parent so that they can survive in local environment. So we have this problem now, okay? Because 50% background gene from New Zealand, 50% from Malaysia, so cannot. So what must we do? Okay, background genes are genes that code for adaptive features of organism in a particular environment, so they can survive in this environment. So how do? We do back crossing. Back crossing will resolve this issue. Okay. Back crossing is a way to increase the background genes of the local parent in the offspring. It's a method for us to increase the background genes of the local parent in our offspring so that it can survive very well in this environment, so that the superior gene can be expressed. Okay, so how do we do that? Back cross means what? You cross backward. So you take the offspring. In every generation of the offspring, you take offspring and you back cross it with your original local parent to increase the percentage of background genes from the local parent. Okay, so we do the calculation, then you understand. So we need to do 5 to 6 generation also. So we have got P1 crossed with P2. These are two superior parents. So let's say these two superior parents is carrying small r, big R, homozygous pure breeding, contrasting characteristic. So then they produce an offspring F1. Now F1 is carrying how many percent of P1 gene and how many percent of P2 gene? Class? How many percent? 50, right? Because when P1 produces gametes, each of the gamete will take 50% of their chromosome and therefore 50% of the gene, correct? Hello? Homologous chromosomes separate, right, into different gametes, right? Each gamete will take half, isn't it, of total. So take half of total chromosome is to take half of total genes. You take half of total allele. So P1 will be carrying 50% of the, F1 will be carrying 50% of P1 genes and 50% of P2 genes, correct? I don't need to write percent. Okay. You all check whether you need to write or not. No need. No need to write. So good. Oh, okay. Okay. So now for F1, I want to increase the percentage of background genes from P1. So what I do, I take F1 and I go and cross it with P1. So when I cross it with P1, I know that from F1, right, when they produce gametes, what happens is that here from 50%, the homologous chromosomes separate, right? Each gamete will take only 25%, correct? Here also same thing. Homologous chromosomes separate during meiosis, so each gamete only take half of it. So half of it is how many? 25%. Right? So each gamete only carry 25%. So therefore, my offspring as a result of the back cross is called BC1, means back cross number 1. Okay, my offspring that's produced from the first back cross is called BC1. So how many percent of P1 gene does it have? So P1 gene, it actually inherit this one, isn't it? Okay, inherit this one from F1 because separate into gametes, so half. So this one comes here. And this one is from P2 comes here. Okay. Then for this parent, out of 100% of the allele or gene, during meiosis, homologous chromosome separate, so becomes 50% in each gamete, correct? So each gamete only carry 50%. So your BC1 only get from one of the gametes, correct? 50%. So therefore, this 50% will come here. Sorry, where is it? Here. 50% will come here, right? So therefore, total how many percent here? Plus? Hello? 75%. And then how many percent P2? 25%. Correct? Okay. So can you see immediately we have increased the percentage of P1 gene already? Can you see not? This is 75% and P2 now is 25%. But this is not enough. So we continue with the back cross for 6 to 7 generations. So now can you please fill in the blank? Fill in the blank for the rest of them and then check answer. I give you 2 minutes to do this. Okay, so please check answer whether you can get the answer or not. So when you continue to do the back cross, you can see that the percentage of background genes from the parents actually increases, right? All the way until when you go to BC6, that means the 6th back cross generation is already 99.22% of the background genes from parent. So this cow can survive in local environment very well. So this is how we use back crossing to dilute the genes that come from P2 by increasing the proportion of genes that come from P1. So it's a very wonderful technique, but very tedious, right? Can you imagine how long you need to wait for this to happen, right? It's very long, very tedious. So now the solution is use genetic technology. Just cut and paste. Clear or not? All right, so this was last time. This one is a very powerful technique, but now with genetic technology is even more powerful. Okay, it's faster, some more specific. Okay, now this table shows you definition of certain term. So if you're interested to know the meaning of the term, you can refer to this table. Some of them are not in a syllabus, just for your interest only. Okay. So let's put together the steps in artificial selection to recap this topic. Human act as selective agent or selection pressure. Human choose both parents with desirable characteristic, and we cross the parents to produce the offspring, and we select the offspring having both the desirable characteristics of the parent. Repeat the selfing and selection over 6 to 7 generations. I cannot say selfing lah. Selfing is for plant, isn't it? So I should say repeat inbreeding, isn't it? Repeat inbreeding and selection over 6 to 7 generations for animal to increase desired allele frequency and homozygosity in the population. I can do a back cross to increase the background genes frequency from one of the local parent so that the offspring is better adapted in the local environment. And then after that, I must do an outbreeding enhancement to overcome inbreeding depression. So therefore, this is all about the artificial selection. So we have looked at the principle of selective breeding, and we have outlined the 3 examples of selective breeding, right? Disease resistant in wheat and rice, inbreeding followed by hybridization to produce vigorous uniform varieties of maize, and improving the milk yield of dairy cattle.